Okay, good morning. So today we're going to talk about a very important phenomenon in genetics, that of complementation. And I'm going to start with one example of deafness in two families to illustrate the point. Okay, so we've got a pedigree here, two pedigrees really, where we have two families and we have some deaf children in the second generation. Okay, and this is both are autosomal recessives, if you look at the people. And we have then in generation 2, person number 27 marries person number 28, and both of those people are deaf, and yet all of their children have normal hearing. So the question is, how can we explain a phenomenon like that? Well, the answer, of course, is complementation. Okay, the idea here is that any phenotype, such as hearing, which is an incredibly complicated process, has many, many genes that contribute to it. So if we have many, many genes that are all necessary for normal hearing, then mutations in different genes can certainly occur. So that's what's happened in these families. The parents, the original grandparents, they have mutations in different genes, both of which are needed for normal hearing. And the combination in the last generation is because whatever was wrong in the family on the left and what's wrong in the family on the right were two different things. So the combination is put together to give normal hearing because one wild type made up for the defect in the other mutation. All right, so the idea is clearly many genes contribute to any phenotype, but conversely, and we'll see this in our analysis in a little while, mutations in many different genes can produce the same phenotype. And so one goal of mutation analysis, or creating mutations even, is to find out how many genes are involved in any process, to identify them, and then study them to understand what their roles might be. Okay, so in the case of deafness, if you look at online Mendelian inheritance in man, you'll find many, 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 many different genes that have been identified as being necessary for normal hearing. And clearly the parents in our pedigree had mutations in different genes. Okay, so the idea, of course, these are going to be recessive mutations, and if you look at the pedigrees, you can see that they're autosomal mutations. And okay, so when we look at that first generation and we try to figure out what's going on there, how is it possible that the two of their children get married and have normal hearing, we can decide that the genes involved in the two families are different from one another. So the two sets of parents in the original generation were both heterozygous for a gene required for normal hearing, but the two genes were different from each other. Okay, so the only options here we have are big A little a here and big B little b here in C, right? The idea is that the parents were themselves heterozygous for a mutation which would cause deafness, in this case the little a, in this case the little b, which explains why some of their children were in fact deaf. As an autosomal recessive, if both parents are heterozygous, some of the children will be homozygous for the recessive trait. But we have to put in a second gene over here because of the fact that when the two deaf children in generation 2 got married, their children were all of normal hearing. All right, so the idea, of course, is that both of the parents were homozygous, or heterozygous rather, for mutations that cause deafness, and they had deaf children as a result. But we can see that the genes are different because when the children will have normal hearing. Okay, so the two deaf parents in the second generation are little a, big B, or both, or little a, big B, and big A, little b. Well, the answer is B, clearly, that the wife was deficient or recessive for one mutation which was required for normal hearing, and the husband had the mutation in a different gene. So they're both deaf, but for different reasons. All right. So we can certainly see when we look at their children what's happening, that mama was heterozygous, or mom was homozygous for little a, dad was homozygous for little b, and when we put those two together, we get heterozygous for both genes, which is big A, big B, and have normal hearing. So that's the explanation for having all these normal hearing kids from two deaf parents. Right? So the mutations complement each other. Each of the children got a wild type allele of both of the genes, one from one parent and the other from the other parent. So the result is they are normal hearing because they have a wild type allele of both of the genes that were involved in this process in the first place. Okay, so it's often the case when people want to study a process or phenomenon in the lab, what they do is they want to have mutations that identify the genes that are involved in those processes. The idea, of course, is that if you need to create these mutations in the lab because they don't exist anyway, and if you have mutations that are defective in a process, the genes that are defined tell you what genes are important for that process. So the way this is typically done is organisms are treated with chemical agents, mutagens, that are used to create mutations. Then the survivors are examined to try to find mutants of interest. So the idea is to find the genes of interest that contribute to the process under study, whatever it may be, and we do that by mutating them and seeing what goes wrong. So here's my fake flower color example. It illustrates a simple fake pathway where we have a precursor that is converted by means of enzyme 1, encoded by gene A, to one intermediate. The intermediate 1 is acted on, again, by a second enzyme B to produce intermediate 2, and then intermediate 2 is acted on by a third enzyme, enzyme C, to produce a product. Now in my fake example, I'm going to say that the precursors, intermediate 1 and intermediate 2, are all colorless compounds, and that the product of this series of reaction is blue. And we're going to say that the blue color is the color of the flowers that are produced by the plants under normal circumstances where this pathway is operating properly. Okay, so mutations in genes A, B, and C, because they're upstream of the blue color, will all produce the same phenotype. If we mutagenize gene A to be little a, or little b, etc., the flower will be white versus the normal blue ones because the blue compound cannot be formed. So the question is how we could use mutant analysis to find this out. So we're first going to get mutations that have white flowers by mutagenizing, looking for survivors and doing a lot of crosses to get them homozygous. And then we're going to compare them one to each other in pairwise combinations in something called complementation analysis. All right, so we're going to mutagenize, get white plants, and in principle, if we get a whole bunch of white plants, we will get mutations in these three genes, all of them. They all create the same phenotype, and so the flowers will look the same, but we realize that those mutations will define 3 different genes, not just one. It's just that the phenotype is the same. So we need a way to sort out which mutant is the same as which, and which one is different. All right, so we're going to cross them in pairwise combinations and look for phenotypes. So if you put the white flower mutants together and you get a wild type, that tells us, assuming that the mutations are recessive, which we will assume for now, if you put them together and you get wild type, that tells us that the mutations are in different genes. That is, complementation occurs. If you put them together and you still get white, that tells us that the mutations are in the same gene. They do not complement. So we construct what's called a complementation table to put the information together in a way that's visible and easy to look at. All right, so here's my complementation table. The mutant strains or mutant plants are I, J, K, L, and M. And we're going to make a table in which we're going to fill in the squares according to whether the phenotype of the cross, when you put those mutants together, is going to give you the wild type or the mutant form. Now, if you see a minus here, we have I and L. We can see that I and L do not complement each other. So that tells us that the mutations in I and L are the same as each other because when you put them together, the organism is still mutant. If we look at I versus J, K, and M, well, those are all wild type. So what that tells us is that IL complementation group is different from J, K, and M. So I and L define a mutation or a gene which is different from those involved mutant in J, K, and M. So continuing through the table, looking for other minuses, we can see that K and M do not complement. There's a little minus in the square, which tells us that K and M are mutations or have mutations in the same gene as each other. So I and L are one complementation group, K and M are a second complementation group, and J seems to complement everything, so J is different from the others. So these mutants have defined 3 genes for us, one defined by mutants I and L, K and M, and J. Well, conveniently, we know that there are 3 genes involved in this process, or we're told that from our pathway. So it's likely that the plants that we get will have those are the 3 genes that we found in our mutant hunt. So we cross them in all pairwise combinations, and we have basically 3 genes, 3 complementation groups that we can figure out. Okay, so this summarizes the results from what I said before. There are 3 genes in total. Okay, so you can't really decide which gene goes with which enzyme, where in this pathway. So you have to, genetics can tell you what genes are involved, but you got to go a little further into the biochemistry to figure out which intermediate goes with which and which reaction goes with which particular gene. So we can somehow decide using complementation which gene is which based on the biochemistry that we're not going to talk about in principle. So when we can write crosses among the mutants, we can just discuss them in pairs. All right, so writing crosses having to do with this. Again, this is like the deaf people. The phenotypes are the same. I and L give white flowers, J, white flowers, KM have white flowers, and those define genes A, B, and C for us according to the fake pathway that I wrote. So if you want to make crosses among these, you have to consider that we're not just doing little w times little w. We have to distinguish which of these genes is actually involved in the cross. Okay, so for example, white plant I times white plant K. We think white plant, we're going to define white plant 1 as being little a, and white plant K as being little c, assuming that those are the correct enzymes in that pathway. So when you write a cross like that, you have to put in the other G. You can't just write little a times little c, in order certainly to explain why the phenotype of the progeny is wild type. So we write this cross as little a, big C, times big A, little c. Right? So the idea is that the little white plant, little a, has alleles of gene C, and if you're going to cross with a plant involving gene C, you have to put in the wild type alleles. So you have to have the alleles for both genes that are under consideration in writing any cross. So when it's written this way, you can see very clearly why the progeny are blue and wild type in phenotype, because they got the big A from the K plant, which was deficient in C but not A, and then it got the little a from the plant number I, which is little a, and conversely, it got the big C from the white plant I, which is little a, but normal for C, and it gets the little c from the K plant, which was deficient in gene C. So the progeny are clearly going to be wild type because the mutants are in both genes. Okay, so running this in reverse, if we have a bunch of mutants, how we make a complementation table, you don't in principle have to make a complementation table. You can just write out mutant 1 times mutant 2, mutant 1 times mutant 4, and write down the phenotypes. But this gets very ungainly to look at if you have, in this case, only a couple genes, but if you have 20 or something mutants, you want to have a more systematic way to look at them, and that's why we draw complementation tables the way we do. So this is, I call our mutants in Drosophila. The wild type is a nice dark red color, and this mutant, which is, we're going to call a mutant, is purple eye. So the purple is the mutant. Right. So we're going to draw the complementation table. We're going to call the mutants 1, 2, 3, and 4, and 1, 2, 3, 4. And we're just going to fill in, with the information we have from the above, which ones, assuming they're recessive, which ones complement each other and which ones do not. Okay, so I fill them in. Mutant 1, of course, everybody times themselves is negative, clearly, because you get a little a times little a, it's still mutant. What's important is that pluses and minuses when you're crossing times another organism. So here we have mutant 1 times mutant 2, which gives wild type. So that says those are different genes. Times mutant 3, it's minus. So those says mutants 1 and 3 are a mutant in the same gene. Mutant 1 times mutant 4 is wild type, so that's 1 and 4 are different. If we cross mutant 2 times 4, we find that it's negative, so that means that it's still mutant. So mutants 2 and 4 define mutations in the same gene as each other. So we've defined two genes here, mutants 1 and 3 making up one complementation group or gene, and mutants 1 and 3, and then 2 and 4 making up the second complementation group or gene. All right. So we sometimes show the whole chart filled out. It's sort of annoying to see you get half a chart, but that's because the information within it is redundant. If you look at the chart itself, you can fill them all in if that makes it easier, but actually it kind of confuses the situation, I think, sometimes. All right, so here it is all filled in. Again, we're assuming all the mutants to be recessive, and that everybody is crossed in pairwise combinations, but now we've filled in everywhere. So the original showing of this has, say, mutant 1 times mutant 3 in the top line, but that information is also present as a column if you look at mutant 1 column, read down to mutant 3. So with this way of showing the table, all the information is present twice, whereas a more customary way of showing them fills in only half the table, which is supposedly makes it easier to look at. But if you like having it completely filled in, that's fine too. It's just that you get each cross present twice in the table. Okay, so again, which gene we're talking about and how you write these crosses. The cross between mutant 1 and mutant 2. Now remember, those are different. 1 and 3 were the same, and 2 is the same as 4. So we have to write two genes in here because we're considering two genes in predicting the progeny. So if we write the cross between mutant 1 and 2, call mutant 1A and mutant 2B, then we have to write both genes in the cross. So the answer is D. We have a, big B, times big A, little b, in order to recognize the fact that we have two genes involved in our cross in order to predict our progeny phenotypes. Right. So the progeny are wild type because the mutations complement each other. The mutations are in different genes. Okay, so now we're going to look at an interesting and important example of complementation analysis in people, and this is going to use cell lines to assign complementation groups to mutations causing the genetic disease xeroderma pigmentosum. Okay, this is a particularly devastating disease. There are 7 different genes that can be mutated to lead to this disease, which is the same but variable severity in the different cases. But the whole idea is in this process, which is nucleotide excision repair, there are at least 7 different genes that are necessary for that process, and a deficiency in any one of them will give, if not exactly the same, very similar phenotypes. So these genes encode proteins that function at various steps in this form of DNA repair in response to UV light. Okay, so this is a human genetic disease that leads to extreme sensitivity to UV light. People with XP can't tolerate the UV that's present in normal sunlight. So people have to take extreme protection from sunlight because they often get skin cancer at very early ages. In fact, there's special camps for kids with XP that they do all their activities at night so they can have a normal childhood camp experience despite their problems with their disease. Okay, so here's Camp Sundown, special camps. Right. I didn't want to show pictures of kids with XP, but if you want to see some, look at Google. You know, it's very sad. The kids get skin cancer before they're 20, often sometimes younger than 10. Okay, so the problem here is nucleotide excision repair, which is defective because the various mutations inactivate various proteins that are necessary for that process. So remember UV light, it causes thymine dimers, and these are repaired by the nucleotide excision repair system. So in the absence of the repair system, the DNA damage is not repaired, and you're going to have mutations in broken chromosomes, and cancer does result from these, ultimately, these defects. Okay, remember what a thymine dimer is. Here's a representation that adjacent thymines within the DNA, when exposed to UV light, can form this covalent structure in which the two bases are actually physically linked to one another, right? So this causes a problem when you try to replicate past it and causes ultimately DNA damage. Here's another thymine dimer, slightly different representation, but the idea is the same, that the adjacent thymines become covalently linked to one another, and this causes problems with replication. All right. So we fortunately have a very efficient nucleotide excision repair system, which is able to deal with this form of damage and repair it. Now, XP people are lacking one of the enzymes that are necessary for this process. So as a result of this defect in DNA repair, it doesn't occur. And of course, the skin is especially sensitive to this because it's the skin that's exposed to UV light primarily. All right. So an assay for this process is that normal cells can repair UV-induced DNA damage. XP people cannot do so. As a result, cells from XP people are killed much faster by UV than normal. Okay, so you can use a sensitivity assay, which we'll look at again later, where we're taking a bunch of different cells. The wild-type ones are shown in dark color, and then deficient ones are shown in the lighter circles and squares. And what we're doing is we're taking the cells, exposing them to increasing amounts of UV light, and seeing how long it takes them to be killed. Now note the log scale here. The 100% is at the top, then you have 10%, 1%, and 0.1%. So very, very clearly, the open circle cells are dying off at a very much lower amount of UV dose than the wild type. And this kind of assay can be used to assess repair efficiency, as we'll see in a little bit. All right, so the functions of the various XP proteins and what they do, you don't need to know this, just be aware that the different steps in the process require different proteins that are encoded by different genes. Okay, but if you think about what's happening with DNA repair, here's represented here. First of all, you have to recognize that the damage exists. The little red square at the top represents the thymine dimer, and it's shown as a little bulge in the molecule that has to be recognized, and there are certain XP proteins that are involved in this process. Certainly XPE, XPC, and also people believe XPA are involved in that first stage in the recognition process. The next thing that has to happen is we need the activity of two helicases encoded by XPB and XPD, and what those two helicases do is open up the molecule to allow access ultimately to the DNA repair polymerases. But your first step is to open up the molecule to make it accessible. Okay, so the important thing is those are dedicated helicases for this process. So after that, we have two dedicated nucleases encoded by XPF and XPG, and those nucleases cut the DNA on either side of the damage, one for each side of the damage. Okay, so that's represented here, that we have the damage. We've opened up with the helicases, and then we're able to then chop the phosphodiester bond on either backbone, on either side of the opened up area, and that piece in the middle is actually excised in the form of an oligonucleotide to remove it so that it can then be replaced by repair DNA polymerase. So once that has been accomplished, a repair polymerase will then synthesize the other strand, or synthesize over the region that's been removed, and that will be ligated to restore the integrity of the phosphodiester bonds, etc. Okay, so the point here is it's a complicated process involving multiple proteins, each of which serves its own unique role in that process, and each of which is required for it to go correctly. So mutations in any one of those genes is going to give you deficient nucleotide excision repair, and then the disease, which is XP. Right. So this is an important process, right? All right. So these proteins and complementation groups, which is why we're talking about this example today in particular, XPA through XPG encode different proteins clearly. So a person who is homozygous for XPA, for example, is deficient in that protein, but all the rest of their proteins are okay. Similarly, anybody that's deficient for any one of the other proteins is deficient for that one, but it's okay for all the rest. So these genes are called complementation groups, and we can see what happens to them when we put them together in cell lines or in people, as the case may be. All right. So anybody who's homozygous for any one of these, their cells are sensitive to UV, and they will die if exposed to UV in the assay system we showed before. So what you can do with the people's cells is expose them to UV and see that they lose viability over time, as expected for people that can't repair their DNA. Right, so here's our UV sensitivity assay, and the normal cells survive pretty well, and XP cells are killed off at a fairly rapid rate, easily distinguished by how long they survive. All right, so this is where things get a little weird. We're going to use cell lines from XP people to assign complementation groups. So now we have, these are cell lines that can be purchased, they're available, and they're known cell lines which are deficient in XPA, B, C, D, E, F, G as a group, right? So initially, these were done on a case-by-case basis, but now we have known cell lines that can be used to test people. So this is where it gets a little weird, that to do complementation analysis with cells, you're not going to mate the cells. What you're going to do is fuse the cells and look at the properties of the fused cells to decide whether complementation has occurred or not. So in this example, we have a repair deficient cell with clear white nuclei, another repair deficient cell with dark nuclei. Okay, and that distinguishes one genotype from another. Let's say this repair deficient one on the left is little a. It's deficient in XP protein A. And the one on the right is deficient in XP protein C, for example. We can fuse those cells together to make heterokaryons. That's what we call it. If a cell has nuclei, two different nuclei, or more than one nucleus of different genotype, they're generally referred to as heterokaryons. Okay, so what's being tested here is whether the mutation on the left is the same as the mutation on the right, or if it's different. So if we fuse them, and the mutations are in the same gene, then they're still going to be deficient. That would be, say, little a, little a, little a, little a. You're still not getting any wild type big A from anywhere, so it's still deficient. Okay, so what is of interest here is if the two mutations are in different genes, if you take one that's UV sensitive because it's deficient in A, and another one that's UV sensitive because it's deficient in C, and put them together in the same cell, well, now you can repair your DNA because you've got wild-type alleles of all of the genes, so complementation occurs and the phenotype is normal. Okay, representing that, if I have cells from an XPA person, okay, we're going to make tetraploids. This looks weird, but there you go. So the XPA person will show as little a, but they're normal for all the rest of the genes, so we're going to be considering the XPC person, so we'll write in the C. So they're little a, big C, and cells from the XPC person are big A, little c, little c. So the XPC person has two defective alleles of XPC, but they're wild type and normal for all the rest of the genes. So we have to write in those normal alleles when we're considering both of them together in this sort of cross. So you put them together in the same cell, and you end up with a tetraploid, clearly. Two little a's, two big C's, two big A's, and two little C's. So overall, you're big A, little a, big C, little c, little c. So even though it's weird that it's a tetraploid, you do have wild-type alleles of both genes present, and therefore complementation occurs, right? And the cells are normal. They're resistant because they have wild-type alleles of all of the genes that are involved. Okay, so in terms of complementation tables, normal sensitivity means if you put them together, that the genes are different. If you fuse them and you get a mutation and you have still deficient, that means that they're the same gene. So it's again the same plus and minus situation we were using for the complementation tables as before, just with a few more genes in it. All right, so there are permanent tester lines that are used from all the XP types, XPA through XPG. There's actually a new one called XPV. And so new cells can be tested by fusing to all of these one at a time. So everything should complement except the one that matches a mutation in the cell line. And we'll see that in a second. And complementation, again, is measured by our ability to survive the UV light. All right, so here's a complementation table. And I know the little pluses and minuses are very small. But what we're looking at here is we're not crossing people all together. We're not crossing every possible person with each other. We're taking the standard XP cell lines, deficient in A, B, C, D, E, F, G, which is to say all of the genes, one at a time, are deficient, and fusing them with cells of XP people, 1, 2, 3, 4, 5. So the idea is if, for example, this person 1, his cells are fused with the mutant A, and you get a wild type, we know that his mutation is not A, because his genotype or his DNA or his cells were able to complement the A. So the A, that tells us that is not the problem that he has. Similarly, we fuse them with B, and it's wild type, so he's able to complement that B mutation. So his mutation is not in B. If we get to the C, however, his cells cannot complement or cannot restore normal UV sensitivity or normal UV resistance to the C cell line. So his cells are deficient in C, just as is the cell line. There is no complementation. So we would say, okay, that person's mutation is in XPC. That is where his mutation is. And similarly for all the others, he complements all the rest. So the one that this person's cells do not complement, that's the one that's the disease that person has. Right, so I went through and I circled all these. So person number 1 is deficient in XPC. Person number 2 is deficient in XPB. Person 3 is deficient in XPF. Person 4 is also XPB. And person 5 is XPD. So each person should have a deficiency in one, not more than one, of these particular genes, and we can tell which it is by its inability to complement that particular mutation. All right, so this is what I just said. Now I wrote here, please write the full genotype of the cells that were formed by fusing the XPA cell line and person 1. Now to do that correctly, you would probably have to write the little a's, but also the big for all of the others, FG, A, B, C, D, E, F, G. You don't have to do that, but just recognize the fact that when you're mutant for 1, you are wild type for all the rest of the genes involved in the process. Okay. So if person 1 marries person 3, will their gene have XP? Yes or no? Well, the answer is no, because person 1 has X. They both have XP, obviously, but person 1 is XPC, person 3 is XPF, and of course the XPC person has normal XPF and all the rest. XPF people has normal C and all the rest. So if you write the genotype of the kids, they're heterozygous for those 2 genes, meaning that they've got a wild type for each one. So the kids are big C, little c, big F, little f, and they have normal DNA repair. Right? This is a classic case of complementation because the genes that are causing the disease is different in the 2 parents. Okay. So we're not fusing with cell lines this time. We're going to fuse cell lines from one person versus another, just as sort of an exercise. Since we already know now what their genotypes are from the cell line comparisons, we can clearly say what phenotype you would have if you fused the cells from the people in all pairwise combinations, now that we know what deficiencies they have. Okay, so person 1 is going to complement everybody else. Person 2 is going to complement all except person 4, because they're both XBB. Person 3 complements everybody except themselves. person 4 also. Okay, so the idea is here, of course, that all these people, except for 2 and 4, have different XP variants or mutations in different genes that give XP, and therefore they will complement everybody else's except their own or anybody else who happens to have the same mutations in the same gene that they have.